All right, in today's video lesson, we're going to be talking about what are called the 4 principles of experimental design. Let me kind of set the stage for what we're talking about today. Most of what we've talked about so far has been about observational studies. We've been talking about all the different sampling techniques you can do, whether it's a simple random sample or a cluster or a stratified sample. And we've talked a lot about observational studies. Well, in the last section, we just talked about what the difference was between an observational study and an experiment. And so now we're going to more solidly describe what exactly an experiment is by talking about these 4 principles of what make experimental design. So let's talk about those 4 different principles. The first is what's called control. The second is that we're going to have some form of randomization. So we are going to randomize. The third is that we need to be able to replicate. And the fourth is that we need to consider using some form of blocking. So let's go into a little bit more detail about what each of these is. To start off with, control. When you're looking to establish control, what you're trying to do is you're trying to control sources of variation other than factors that we are testing by making conditions as similar as possible for all treatment groups. To give you an example, I think I mentioned that I had done an experiment, a science experiment, where I had tried to grow grass using a few different liquids. I had used water, milk, and Gatorade. Okay, now that obviously is going to be different for each group, but other things I wanted to keep the same. For instance, I wanted to make sure I used the same soil and the same type of grass seed and the same amount of light, and so on, so that I could truly say that any differences were solely based on the type of liquid being used. In other words, if you didn't keep the amount of light the same for each of them, then you really can't say whether the results are because of the light or because of the liquid. And so it's really important to make sure that everything else besides what you are wanting to test, all those other factors are the same. That's the idea behind control. The second is that we're going to randomize. This allows us to minimize any effects of unknown or uncontrollable sources of variations. And it allows us to begin establishing a cause and effect relationship. Here's kind of the idea of randomize, is that even though you're trying to keep everything the same, there are going to be these slight differences between groups. But you can really minimize those differences as long as you randomize. So in other words, once I had kind of set up my different plots of soil and grass seed and all of that, I randomly chose which one would get water and which ones would get milk and which ones would get the Gatorade. That way, if there were any small differences, the randomization kind of accounts for that. And we'll talk more about that and how it works in a couple examples later on. The third principle of experimental design is replication, or to replicate. So when we replicate, we need to apply each treatment to a number of subjects. And so that was where, rather than just having, a little bit of soil and like one seed, I had quite a few seeds in each group. And so we're applying it not just to one area of this ground and this grass, but applying it to a few different groups and a few different places of ground and grass. The other idea of replication not only is that you're doing it for multiple subjects, but you should be able to repeat the results later on. So you should be able to take a different population, different experimental units, and come up with similar results. So maybe not just looking at the ground and the grass at my house, but maybe going to my neighbors, or maybe going to a friend or family members and doing the experiment with them as well to make sure that it wasn't just because of the grass and the ground at our house that we were living in. I don't know how happy they would be about that, but, it is what it is. The last is blocking. So blocking is where we group similar individuals and then randomize in order to eliminate some variability. This is kind of similar to doing a stratified sample when you're doing a survey. So maybe you already know that one kind of piece of ground does not grow well. Then what you might want to do is separate the ground that grows well from the ground that doesn't, or that does grow well, and then randomly assign a few different treatments to the one that grows well and a few the one that doesn't grow well. That way you can compare between the groups. One way that we block oftentimes is by sex. And so if you think about it, two sexes, male and female. Again, we won't get into gender because there's too many to compare at this point. But in terms of just sex, there's male and female. Now, again, there's transsexual that are going back and forth. We're not going to get too much into that, but let's consider just male and female. Then maybe we want to compare the differences in standardized test scores. So maybe we're going to set up one group as going to be male and one group as female. We're going to give the male group, two different tests, female group, two different tests, so that not only can we compare the different tests, but we can also compare with the sex of the person taking the test as well. So blocking is kind of a way that if you know there's going to be variability, that you can kind of use that to your advantage to get some additional comparisons. Tell you what, let's take a look at an example. So we've got a student who's preparing for a triathlon. They suspected that the 45 minutes spent each day that she spent training in a chlorinated pool was going to damage her nail polish, which if this was my wife, definitely she would be concerned about it. So she wished to investigate whether the color of nail polish might make a difference in how well it stayed on. So how would you design an experiment to test whether or not two nail colors resisted chipping differently when challenged by soaks in chlorine water? And we're going to talk about the 4 principles of experimental design for this particular example. So our first one is control. We need to talk about kind of what we would need to control in this experiment. Well, the first thing that I can think of is the amount of polish that you use. You obviously want to use the same amount of polish on your nails each time because you don't want to really layer it up on one and then leave it kind of thin on the other. Which also means that the amount of chlorine probably needs to be controlled as well. You don't want for one batch to just wear off really quick because there's a lot of chlorine. Some other things that you might want to consider is the amount of time that you spend soaking, making sure that that's the same. You might also want to keep in mind that they are different nail colors. You probably want to use the same brand, and so on and so forth. Lots of different things that you can probably think of as far as wanting to control in order to make this experiment work. So let's talk about randomize. So when we randomize, we can do a couple of different things. We can probably do different random colors. So randomly select the 2 colors that you're going to use. One thing that you can always do when you're randomizing is you can randomly select the order that you soak in. In fact, that's a big one, is that the order that you go in should be something where it's going to be variable between the different times that you do it. So maybe you use one nail polish color first this time, but then you use it second the following time, or vice versa, and you kind of alternate when they get to go first versus second. Next, let's talk about replication. So replication, we want to do this test multiple times. So multiple times, meaning not just soaking one color once and the other color once, but doing that and repeating it. Now, part of replication is to also include your procedures. So writing down procedures will help to make sure that for future experiments, if someone else wanted to also compare nail polish color and how well it stays on, that they could read your procedures and follow it step by step and get similar results to what you got. Now, in this case for blocking, I really don't see a need to block. I guess one thing you could do, if you did want to block, is you could block by hand. So making sure that, left versus right, that the left hand gets, both treatments, the right hand gets both treatments. That way it's kind of eliminated as a variable. That's one thing I could see as blocking. So let me go ahead and say that maybe we do need to do that. But by hand, that's about the only way I can really think that you would block in this type of experiment. Now, once you have the results, how do you know if they're significant or not just due to random chance? And so that's the idea of what's called statistical significance. So when the results that we observe between treatment groups are as large as we would expect, just based on pure randomness, so just based on the fact that they just happen that way, then we're going to say that they're not significant. But if the difference is larger than expected, we're then instead going to say that the results are statistically significant. So the larger the difference, the more significant the results. In fact, we're going to be spending some time in future units talking about how exactly do we know when we have statistical significance or not. So for right now, I just want you to understand the idea of it, not necessarily how to find it. So now that we've studied both surveys and experiments, you can probably see that they are kind of similar to each other in a lot of ways. So what exactly is the difference between the two? Well, that's what we talked about in the last section. So in the last section, we discussed the difference. Really, it came down to whether there was treatment that was being imposed, meaning that you were actually forcing something to happen that wouldn't normally happen, or not. Now remember, if treatment is imposed, that's what we've been talking about today. So that would be an experiment. Whereas if it's not being imposed, that's what we talked about more last time, which is an observational study. All right, that's all I've got for you in experimental design and those 4 principles. In a next lesson, we're going to be talking about a little bit more with experiments, mainly what are the control treatments, a little bit more about blocking, as well as what are called confounding variables. Until then, have a good rest of your day.